Good afternoon. Thank you for allowing me to present my work. I'm going to talk to you about combination of two techniques, stimulation with a microelectrode array and intrinsic optical imaging to investigate local connections in the brain. So we all know that different parts of the brain do different things, and we have pretty good maps, especially for sensory areas of the brain. So, for example, we have the famous homunculus for the motor and the sensory areas. We also have written topic maps for the visual cortex. We also have auditory maps and so on. However, obtaining these maps was not easy. It took many years, and it was mostly done with single electrode mapping, inserting electrode one point at a time and either stimulating or recording. And so, for example, the famous homunculus that we have all seen in textbooks is a cumulative map of 163 patients, and some of these patients have been stimulated 800 times. So it is very labor intensive. Well, now that we do know where the different areas are, and we'd like to know, well, how are they connected to each other? And this is basically the basis behind the Connectome project. But doing so, one electrode penetration at a time is not practical. So, for example, here I have an image of an injection of a tracer into the visual cortex. And what we see is that it is connected to multiple other cortical columns. And so what we would like to be able to see these kinds of connectivity patterns in vivo. And doing this with just inserting electrodes would be difficult. Therefore, we propose a solution which is a combination of a multi-electrode array and optical imaging. So the array would allow for stimulating 100 or even more sites with no time between loss between placing the electrodes in different areas. And intrinsic optical imaging allows for visualizing the network activation over a fairly large region on the order of a square centimeter or so with high spatial resolution. And so I have a picture on the right here showing an example of just stimulation with one electrode. And you see the single stimulation side, the black area. This is with intrinsic imaging. And you also see these projections light up around it, which are indicated by the red areas. So the experimental procedure is we perform craniotomy and durotomy in the general area of the macaque motor cortex and then we do sparse mapping with a single electrode to localize the forelimb representation within the primary and premotor cortical areas. And then we implant a 100 electrode Utah array over the M1 PMD region and under a chronic optical window. So here's a figure of the general area that we've exposed, and you can see the array. It's ready to be implanted. And so what we do then is we pause ventilation for a moment and use a pneumatic injector to shoot the array into the brain. And then we place a transparent silicone window over the craniotomy region, and that is shown on the right here. And this is a sort of a profile view of the preparation. We have a plastic chamber that is being placed around the optical window, and this allows to access it at any time in either anesthetized or awake and behaving animals. And once we do that, we obtain a finer map of the forelimb representation covered by the array in greater detail using microstimulation under anesthesia and with recordings in awake behaving animals. And this is a special array developed by BlackRock Microsystems that allows us to both stimulate and record. So then we map local projections between primary motor regions and premotor regions around the array using microstimulation and intrinsic optical imaging. So here are the results for mapping the area using the Utah array. We've placed the monkey under anesthesia and stimulated at 300 hertz. And out of the 96 available electrode sites, about a third of them elicited movements, and all of them were in the contralateral limb. And about 20 of them were predominantly around one joint, but there were also some complex movements. And so here on the right, you can see some somatotopic organization with certain, the wrist is clustered in one area and the digits are clustered in a different area, which is what you would expect from our earlier studies. And then what we did was record array activity during a reach and grab task in an awake and behaving animal. Essentially would present fruit to the animal, and the monkey would reach out, and sometimes it would do it with one arm or the other. And so here is an activity map of the time when the monkey is in the process of reaching out and grabbing, and you can see on the bottom a raster plot for one of the electrodes. This electrode was evoking elbow movement during anesthetized stimulation. And so you can see that during the height of the middle of the movement, there was activation in several regions, and this is the raster plot for that elbow sensitive electrode, and you see that majority of its firing was around the time when the monkey was reaching out. And this is also showing you the activity before the movement and after, and you can see that the array is relatively quiet. And this is consistent from one trial to another. You can see on the bottom a second reach and grab trial, and they look pretty similar. If, on the other hand, the monkey reached out with the other hand, the ipsilateral hand, the array remained quiet. And here I'm showing you the significantly activated electrodes during the reach and grab task. So you see that the bottom part of the array, mostly in the M1, was particularly activated during this task. And this is a result of, I think, 8 trials. So then we proceeded to map local connections using intrinsic optical imaging and microstimulation. And essentially what we do is we use red light. We shine it on the brain, and we collect images which correlate with local blood flow. And figure A right here shows you an image of intrinsic optical imaging before the animal was stimulated. And on the bottom you can see the image at the height of stimulation, and you see several areas light up. And here we show the significantly activated areas. And to orient you better here, the black circle, black square represents the location of the array in PMD, and a little bit in M1, and this is the area that you're looking at. So essentially what we're seeing is that we can stimulate in PMD and observe activation in a different area in PMV. And on the bottom you see the time course of 3 regions selected. 1 and 2 are the regions that are significantly activated, and then 3 is just a random region to show as a control, and it experienced no increase in signal over time. And this is a similar experiment. However, a different electrode was being stimulated. A is just a figure of the exposed brain under green light to highlight the blood vessels. Then B is the intrinsic optical image before the stimulation, and C is at the height of a stimulation. And you can see here that the pattern is different. It's more diffuse, and there's one general region being activated. So there are differences in patterns depending on which of the electrodes is being stimulated. So to conclude, optical imaging and microelectrode array stimulation can be combined to quickly and reliably map local cortical networks. And these networks can be modulated and recorded from both electrophysiologically and optically in awake and behaving animals. So in the future, what we would like to do is to perhaps use transparent arrays so that we can actually see what's going on within the area that is covered by the array, and also to stimulate multiple sites simultaneously or sequentially. And that is it, and I would like to take any questions if you have any.